Good morning, ladies and gentlemen. Before we begin, may I ask you to switch off your phones and to stand up for the academic procession. Good morning, everyone. Good morning, Professor Liz-Marzán. Welcome to Atrium B, this beautiful location in the heart of the University of Antwerp. It's recently renovated. And welcome to the conferral of one of the honorary degrees for 2023. My name is Anke. I'll be your hostess this morning, and it's an incredible pleasure to be here, surrounded by so much knowledge, intelligence, hard work, and talent, of course. Let's make it a warm and interesting morning. Honorary Dr. Liz-Marzán will shortly be teaching a masterclass titled From Nanoparticles to Artificial Tumors, a Lifetime Scientific Journey. During the masterclass, you can ask questions using the chat function on the YouTube channel. Of course, this is for the people who are following this ceremony online. After the masterclass, there will be time for a Q&A session in which Professor Liz-Marzán will answer the questions you submitted. Of course, the people here in this room can also participate in this Q&A session by using the very old-fashioned technique of raising your hand. Then we will proceed to the conferral of the honorary degree, of course, but first I would like to give the floor to Professor Gerrit Beemster, Dean of the Faculty of Sciences, who will offer you all an even warmer welcome.
Dear Dr. Honoris Causa, Professor Louis Liz-Marzán, dear Rector, distinguished colleagues, guests, friends, family, ladies and gentlemen here present and online. As the Dean of the Faculty of Science of the University of Antwerp, it's a real honor and a pleasure to welcome you all today to the masterclass and the official ceremony of the Doctorate Honoris Causa to Professor Liz-Marzán. An honorary doctorate allows the faculty to decorate a really exceptional researcher and to highlight important research in our field. The translated, because I think there is only a Dutch version of it officially, slogan of our institute is that the University of Antwerp helps determining the future. Okay? It's obvious that currently our society is facing an increasing number of challenges with regards to this future. Think about climate change, food security, sustainability. And action is required to secure the future for our following and next generations. As scientists, I think we firmly believe that detailed scientific knowledge of how the world around us works at all levels, from the whole society all the way down to molecular scale, nanoscale, knowing all that offers a basis for developing solutions to these challenges. It is therefore no surprise that finding answers to society's challenges based on scientific insight is actually what connects the past doctorates honoris causas from our faculty for example, Professor Matthias Beiler, who is working on bio-inspired catalyzers for sustainable chemistry. Dr. Christophe Lasseur, who investigates ecological life support system, providing profound insights as a basis for how we should organize a circular closed-loop economy in the future. And the previous laureate, Professor Elaine Lubchenko, is a marine biologist who functioned as a key advisor to ex-US President Obama with regards to climate change. So we are very proud that Professor Liz-Marzán and his eminent work on nanoscale metal particles will be added to this, what I think is an impressive list. At this point, an important thing to highlight is the importance of strong collaborative bonds to do top science. It's needed for the exchange of new ideas, for the training of the next generation of young scientists, and, of course, also to develop successful grant applications. And I just heard that you guys are very hard working on those as we speak. In this context, I must say that an honorary doctorate provides a direct link to our university. And we consider our laureates as important ambassadors to represent our university after they have been awarded, and that they further extend and strengthen our network of international collaboration. Finally, I would take this opportunity to express my appreciation for the promoter of this honorary doctorate, Professor Sarah Bals, who will next introduce Professor Liz-Marzán's career and research in much detail. And I'm convinced that we are going to have a very interesting event and hope that you will all enjoy the masterclass that is going to follow.
With this honorary degree, the Faculty of Science of the University of Antwerp pays tribute to Professor Louis Liz-Marzán for his pioneering work on plasmonic metal nanoparticles. Louis obtained his PhD in chemistry from the University of Santiago de Compostela in 1992 under supervision of Professor Arturo López Quintela on the synthesis of magnetic nanoparticles via microemulsions. Next, he performed postdoctoral work at the University of Utrecht, where he collaborated with Professor Albert Philipse and started a project on the synthesis and characterization of metallic nanoparticles. He then returned to Santiago as an assistant professor, but quickly moved to the University of Vigo. He became a full professor at this university in 2006, and 6 years later, in 2012, he moved to the CIC Biomagune Institute in San Sebastian, where he became appointed as an Ikerbasque research professor and as the scientific director of the institute. The part-time return of Louis to Vigo last year was another important milestone, and one that was even covered in the Spanish press. Throughout his career, Louis has been appointed as a visiting professor at different universities, and he received numerous awards at the highest national and international levels. It is worth to note that he is the most cited Spanish scientist in the field of material science. And for those who like numbers, his H-index equals 150, his work received more than 80,000 citations, and he holds 2 advanced ERC grants. Now, all of these successes are based on the fact that Louis is a world leader in the field of colloidal nanoparticles for nanoplasmonics. He was one of the pioneers in the colloidal synthesis and characterization of these metal nanoparticles. And in particular, he specialized in obtaining great control over the morphology of gold and silver particles of interest because of their fascinating optical properties. However, the work of Lewis went beyond controlling just their size, and he developed novel protocols to obtain almost every shape that one can think of: spheres, cylinders, triangles, even nano-dog bones or nanostars. Most recently, Lewis and his group demonstrated the growth of so-called chiral nanoparticles with world record optical properties. Indeed, at the nanoscale, light interacts with metallic nanoparticles in such a way that specific colors are created. This phenomenon is based on what we call plasmon resonance. And although they were unaware of the science underlying their work, stained glass artists in the Middle Ages were early users of this type of technology. Although I consider Lewis as a true artist in the nano world, his research is going much beyond the creation of beautiful colors. And his work has created diverse and important impact in different fields. Among other things, he has contributed to the design of substrates that improve the performance of sensors based on the concept of plasmon resonance, and as such, these systems can be used as biomarkers. Applications of such technology can be found in pregnancy tests and more recently, COVID tests. Another application of plasmonic nanoparticles is hyperthermal cancer treatment, where the heat that is released after shining a laser onto the gold or silver particles may destroy tumor cells. Recent work by the group of Lewis has therefore also focused on obtaining a better understanding of the fate of these nanoparticles in living cells. I'm standing here today as a physicist, nominating Professor Liz-Marzán, who is a chemist, for an honorary degree. This may sound a little bit odd, but I truly believe that multidisciplinarity is a requirement to make progress in an exciting field such as nanoscience. This idea is also reflected in the research by Lewis, whose ambition is always to perform morphological and optical characterization at the highest level and to apply theoretical methods to model particle growth and optical characterization. Next, these systems should be applied in impactful applications. And all of these aspects are part of the title of the most cited paper of Lewis, which is Gold Nanorods, Synthesis, Characterization, and Application. For all of these reasons, we are deeply grateful to Professor Louis Liz-Marzán for having accepted this honorary doctorate from the University of Antwerp and for giving us a masterclass today. And I would now like to invite Professor Liz-Marzán to the stage to present his masterclass.
Good morning, everybody. It's a real honor to have been nominated for this honorary degree. And there will be time later to acknowledge contributions and thanking the university. Now I have the duty to impart a masterclass, which I hope will give you a glimpse of the type of research that we have been doing over so many years now already. And the title already indicates a little bit of an evolution of our research. In fact, rather than from nanoparticles to artificial tumors, I might have called it from surfactants to artificial tumors, because this scientific journey actually starts with what we call surfactants, which are molecules that are the main ingredient of what we all call detergents or soaps. And these are molecules which contain two well-differentiated parts, one that we call the hydrophilic head group and one that we call the hydrophobic tail. And the special properties of these molecules is that they like water on one end, but they like oils or fat on the other end. And therefore, if they are dissolved in water, they like to associate, as you can see here, forming aggregates where the hydrophobic chains are coming together close to each other, while the hydrophilic head groups are facing the aqueous solvent. And in fact, this is the basis of the way we use them every day. The surfactant in the detergent is forming what we call micelles. They get together, they approach the fat stains that we have on clothes or on dishes, and then they associate with the fat. They form droplets, and this fat is taken away from the stained clothes or the stained dishes, so we are cleaning. Now, during my PhD, I had to learn how to work with surfactants in order to be able to create other types of materials. And so I had to learn about the phase diagrams of these materials, meaning that the association of the molecules not always goes into forming these sort of spherical aggregates, but it depends very much on the composition of the system. They can associate in many different ways, as you can schematically see in this diagram. And in fact, this type of surfactant systems, and especially what we call emulsions have been used for a long time to create well-defined spherical polymeric particles. And the idea is simply that the monomer is encapsulated in a surfactant droplet. It slowly exchanges with the solvent, and then there is an initiator that promotes polymerization into these very well-defined particles where the size is determined by the presence of the surfactant. Now, in my PhD, already 35 years ago, I was studying a particular type of surfactant, and I had to study the phase diagram to then decide at which compositions I would be able to create droplets, in this case opposite to what we are doing usually, so droplets of water which can be dispersed in what we call an oil phase. And the idea was that then we can create this sort of micro or nano, we would say today nano reactors. They will exchange the content of the water pool, and then there will be precipitation of a solid material inside of the droplets, so that their growth will be limited by the size of these droplets. And in that way, we were able to create magnetic nanoparticles or nanomaterials with very special properties. I decided to show you here that actually at that time, we did not yet say nanoparticles. You can read here, it was called ultrafine magnetic particles. And this was the type of electron microscope images that we were able to get in 1992, where we were mainly guessing what we had and estimating particle sizes. In fact, the sizes were more reliable from the magnetic measurements than from the electron microscopy images that we could get. Anyway, so after that, I moved to the Netherlands, and I started actually a project which was related to studying the diffusion of colloids in confined spaces. So I had to learn quite a lot about diffusion, and diffusion which is limited by constrained spaces, and about light scattering techniques and a few other things. However, it was quite coincidental that during this time, not so late after I started, there was a lecture in a seminar in the department which was related to explaining how pregnancy tests work and how they were connected to colloid science, which was the specialty of my department. And so somebody came from the company and was trying to explain how these systems work where we have this stripe which would be immersed in the urine of the woman who was to be tested. And then we would watch after a few minutes for a line here which would indicate whether the test is positive or negative, and another line here which would indicate if the test is valid or not. And during the lecture, this person was actually carrying out an experiment without saying what he was doing. And there was this pot of water, apparently, which was being stirred and heated up. And over time, we would see that the color of this solution was changing and getting darker and darker. After some 15 minutes or so, the end result was something like this, a very beautiful wine-looking solution. And this speaker explained that the reason that we can see the laser beam going through this liquid, which otherwise looks completely transparent, is the presence of very small particles floating around, in this case, particles made of gold. And the reason he was showing this is that actually these gold particles are the secret of how the pregnancy tests work. Because what we have in the pregnancy test is a reservoir somewhere here, which contains gold nanoparticles, which are carrying antibodies which selectively recognize the pregnancy hormone. And so if the hormone is present, which would be represented by these pink stars here, they would bind the antibodies on the nanoparticles, and then they would bind again onto the test line. And then there is a second line which contains another set of antibodies which will anyway recognize the nanoparticles and will tell us whether the particles have traveled far enough through this porous stripe so that the test is valid. And I'm sure that you are today all familiar with these type of tests, because this is what we all have been using during the past 3 years to test the COVID. It's exactly the same concept. So we can say that we were already knowingly using nanotechnology since the 1960s or so, when the pregnancy tests were developed. So after learning about this, I started reading about nanotechnology. I learned about Professor Richard Feynman, who got a Nobel Prize on something completely different, electron chromodynamics. Anyway, in 1965, the year I was born. But he was also a visionary, and he was giving popular lectures, and one of his lectures was titled, "There is Plenty of Room at the Bottom," and there he proposed that if we were to study the intermediate sizes between molecules and the bulk, then we should be able to find completely new properties which would revolutionize the industry as it was known in the 1960s. For a definition of nanotechnology, I like this one, which mentions materials and devices where nanoscale size components is the most important part. And it is important because the properties of those components only arise when we are in the nanoscale. And even more, at this nanoscale, a millionth of a millimeter, if we change slightly the particle size, the properties will also change. And so that makes it a very sensitive type of material to find completely new properties that we can apply in many different fields. And the reason that the nanoscale is important is that when we are reducing the particle size, the proportion of atoms which are at the surface of the material is getting larger than the number of atoms that are contained inside of the material. And so because the chemical properties of these atoms at the surface is different to the atoms in the inside, for very small sizes, surface properties dominate over bulk properties. And again, because this proportion changes dramatically, as you can see here, when we change slightly particle size in this domain, we are changing the properties gradually with particle size. Now, as Professor Valls was mentioning in the introduction, this was actually already proposed for many different fields and known since a long time ago. I'll go into this in a minute. This is a slide which is already a little bit old, and in fact, the original title on the slide was Nanotechnology in the Future. This was less than 10 years ago, and it was predicting that we would be using nanomaterials for preventing corrosion, for making thermochromic glass to regulate the influx of light, for organic light-emitting diodes in displays, photovoltaics, scratch-proof windows, the menu cards made of electronic cardboard, nanotubes for notebook displays. Today we would probably say graphene for displays. Fabrics coated to resist stains, and so on and so forth. These are all materials that we already have in our daily lives. And most of them are based on the properties of nanomaterials. So this has really become a revolution in the way that we live, even if we don't notice it. But again, this was known already thousands of years ago, not exactly that we had nanomaterials. But the Romans were already fabricating these glasses, which contained very small pieces of metal, through which the color of the glass was changing depending on whether it was illuminated from the back or from the front. And this particular cup is called the Lycurgus cup. It's on display in the British Museum, and it's displayed in a shelf where there is a light behind which is switching on and off intermittently, so you can actually admire this color display looking green when the backlight is off and red when the backlight is on. And after extracting a small piece of this glass and looking again in the electron microscope, they found this collection of small particles made of gold and silver, which are responsible for those colors. And something similar happens in the stained glasses in churches and cathedrals and many other old and new buildings, where the colors have to last very long, so they are made of inorganic materials, but still they should provide colors without becoming too turbid, and so these stained inorganic materials have to be very small in particle size. This is all related to the properties of very small particles. Now, the first scientific document that we have describing these properties is probably this article by Michael Faraday. Faraday, whom is best known for discovering electricity, so to say. He was also interested in many other different fields. He actually had a very interesting life, all dedicated to science and to family and friends. So I sympathize very much with him. He was a member of mainly every scientific society in Europe or in the world, including the Academy of Brussels. And he wrote this article after spending 1 or 2 years in his laboratory at the Royal Institution, studying the properties of very thin metallic films and very fine dispersions of particles in the liquid. And so the title of the article is Experimental Relations of Gold and Other Metals to Light. And the reasoning behind the work is understanding the undulatory theory of light. So he was trying to understand what Maxwell was proposing. And anyway, this was done at the time, 1856, delivered, when there was still the belief that the ether was mediating the interaction between bodies. So, in fact, if you read the start of the article, Faraday was not very much convinced that this was the right thing, and he writes, I have often struggled to perceive how far that medium might account for or mingle in with such actions generally, and to what extent experimental trials might be devised which, with their results and consequences, might contradict, confirm, enlarge, or modify the idea we form of it, always with the hope that the corrected or instructed idea would approach more and more to the truth of nature, and in the fullness of time coincide with it. So I think this is a very beautiful definition of scientific work. Anyway, so during this work, Faraday was really making gold colloids, using mainly, but not only, white phosphorus as a reducing agent, and describing the formation of what he called beautiful ruby fluids containing exceedingly fine particles that we would call nanoparticles today. And again, I always recommend everybody to read this article. It's a very pleasant reading, and in fact, you will find there many discoveries that have been rediscovered again and again over the years. If Faraday would have access to the instruments that we have today, he would be really amazed to find what he was actually discovering. He did not have access to any spectrometer, so he could not really decide on data what he was describing as the differences between the properties of the films and the colloids. Of course, he did not have access to an electron microscope, thing that photography was invented less than 20 years before this work. So he was only guessing about the size and the composition of the materials. Now we know that these particles are actually made of exactly the same type of metallic gold as we use in our rings or jewelry. Anyway, so the reasons for the colors that I have just described are surface plasmons. So plasmons are actually charge density waves in materials containing free electrons when they are excited by an electromagnetic radiation. So in bulk metals, you can have these waves traveling through the metal, what we call the bulk plasma frequency. In very thin films, the waves will travel at the interface. In very small particles, at the very limit of very tiny particles with spherical shape, you will have the formation of a dipole. And because we have an oscillating electric field in the radiation, there will be coupling at a certain frequency of the oscillation of the electric field and the oscillation of the dipole, of the electrons in the dipole. And this is what we call the localized surface plasmon resonance. And this is what defines the colors of dispersions of gold and silver. And maybe not everybody is familiar with the fact that in dispersion, gold would look red, silver would look yellow. And I say in dispersion, but I should say in dispersion of small spherical particles, because remember, in the nanoscale, particle size, and you will see in a minute, particle shape, actually determine a lot the properties of the material. Another important property that may become a bit more specialized is that when we are exciting this oscillation of electrons, we are also creating very high electric fields at the surface of the material, which will be important because it can affect the spectroscopy of molecules coming in contact with the metal. And this is very important for applications. Now, when I returned to Spain after postdoc, I had to start a new group, and it was a very recently created university with limited access to instruments and to students. And so we had to rely partly on collaborators. And I wanted to mention here 3 names that in the early stages of my career were very important. Michael Girsik, Paul Mulvaney, and Nicholas Kotov. The collaboration with these 3 people was essential to really set up the path of my research. We came up with this discovery of a method to create spherical gold particles coated uniformly with silicon oxide. And this has been extremely important for many studies and applications everywhere. And as you can see in these pictures here, by controlling the thickness of this shell, we could actually control how far apart the metal particles are to each other. And this actually changes the way they interact with light. After that, and also with, of course, the hard work and contributions of the first students in my lab and the first postdoc, we were discovering more and more of these effects, which, if I'm totally honest, after reading again Faraday's paper last week, I realized that many of these things were already described by him. Only he did not have the means to really show them so beautifully as we can now. Anyway, so through the years, we have studied really the formation of particles with different morphologies, and one of the most controlled syntheses that we have apart from the spherical particles that I showed you already have these dipolar oscillations of electrons, is the creation of what we call nanorods. And these are simply elongated pieces of metal in the nanoscale, which, because of their shape, can interact with light by oscillating electrons along or across the axis of this elongated particle. And because of that, you see here, there are here two wavelengths, two frequencies at which they can interact with light. And so we can have actually two different colors depending on the polarization of light that we have and depending on the orientation of the particles. Through colloid chemistry methods, we can make particles as beautiful as this. And by controlling the ratio between the length and the width, we can modulate the frequency of light that will predominantly interact with them. And this opens up, again, many possibilities to tune the work that we want to do in applications of these materials. Still, if you look carefully, actually, by changing conditions, you can change not only the colors, but also the morphology into different ways. You can create multimetallic particles, and this was already highlighted almost 20 years ago. And still, we are working hard to understand well how these particles form. The method that we use typically comprises two main steps. One is reducing gold chloride by a very strong reducing agent and forming very tiny particles, about 1 or 2 nanometers. You can hardly see them in the electron microscope. And then these we use as seeds for a controlled growth into the morphology that we like. And of course, we have to use different conditions to do that. And then we can obtain this type of particles that for some time we were struggling to understand how they looked from a crystallographic point of view. One of the important components in this synthesis is, again, the detergent, the surfactant that we use, because the interaction of these surfactants with different crystallographic facets also changes depending on the type of facet. And it also regulates the kinetics of growth of these materials. And so after many years of study, many PhDs and many postdoctoral projects, we now have this diagram, which is still hard to follow, where so many different parameters will actually affect the growth of these materials. We could still think that it is remarkable that we are able to obtain uniformly shaped particles by using these methods, because anything you change can change either the thermodynamics or the kinetics of the process, and you can go from one morphology to another. Still, by using not only the experience and the theoretical analysis that we do, but also the characterization techniques, we are now much better able to understand what we are doing. And in fact, I can show you pictures like this, and you can see many rods, but you still don't really see how they look at the facet control at the atomic scale, until we were so lucky to meet Staffan Tendelo and Sarah Bals 11 or 12 years ago. And they showed us how, by using electron tomography, measuring pictures at different angles and then reconstructing in a very careful manner, you can really get the three-dimensional image of these materials. And not only the three-dimensional image, you can make particles where you have a golden core coated by a silver shell, and by their methods, you can clearly distinguish exactly the position of each metallic component, which will, in the end, determine also the optical properties of the materials. We can look at different structures. These ones contain crystallographic defects that can only be properly studied by doing this very beautiful atomic resolution electron tomography, where you can identify precisely the position of every atom in the particle and then decide how it is going to behave. We can also look at these other materials, and I told you we are making the particles by first making seeds and then growing them. Sarah and her team are able to see where the seed is located inside of the particle, and this helps us, of course, to understand how the growth is happening. And so, in fact, with all this body of research, we proposed a few years ago, that in order to really control the growth of metal crystals at the nanoscale, we need to have access to simulation tools, but also the best characterization tools in order to understand. Currently, we are working hard to apply also study of these processes in liquid systems to better understand what we are doing. But anyway, through this collaboration, we were really able to propose the growth of these type of particles, which look very strange to you. They also display very special optical properties. In this case, what we call circular dichroism, and I'm not going to explain that today. But having access to this type of characterization was crucial, and very recently we were able to see also that we can distinguish the position of the central nanorod core from where we are growing these helical patterns that determine the optical response. Now I'm going to go a bit faster because I'm running a bit out of time. So we have this library of nanoparticle shapes, and now what do we do with them? Now, as Professor Valls indicated in the introduction, in 2012 I became the scientific director of a center that is called the Center for Cooperative Research in Biomaterials. And so we had already the ambition to find bio-related applications for these materials. And one of the reasons is that by manipulating the shape and the size of the particles that we make, we can tune their optical response to a specific wavelength range, which coincides with one of these windows where human tissue becomes more transparent. And so now, by designing the nanomaterials that we make, we can go and find applications that can be relevant to use in human bodies. And this is something that opens up many possibilities. You can start thinking about therapy, but you can also think about diagnosis, about understanding biological systems. And so we started to work on this sensing modality, which combines nanoplasmonics and Raman scattering. And so I mentioned already the formation of these high electric fields at the surface of metal particles. If we can make the particles with these very pointy arms, we can obtain even larger electric fields. And this can be combined with this type of spectroscopy that is called Raman scattering, where we can accurately determine the vibrational modes of molecules. We can recognize what we call the vibrational fingerprint of a molecule and recognize it precisely. But in the standard way of using this technique, it's very, it really requires a lot of material. It's not very sensitive. But if the molecules are coming in close contact with our metal particles, then we end up with a modality that is called surface-enhanced Raman scattering, where the signal becomes amplified at the fourth power with the electric field that we are creating here. And so by optimizing the electric field enhancement on the surface of the particles, we can optimize the enhancement of this signal that will tell us precisely whether a molecule is present in our system or not. One way of increasing this electric field is also bringing particles close to each other and forming what we call hotspots. In order for you to understand a bit better how this works, I'm going to show you two videos next to each other, one in the nanoscale, one in the macro scale. And this is what happens when you simulate the interaction of light with two particles in the nanoscale. This is what happens if you put two grapes in your microwave and shine microwave light, so to say. You're creating this interaction between the conducting particles and creating a very hot spot really where they touch. It's exactly the same thing as we are creating in the metal particles, but only creating a very high electric field in this case. By the way, don't do this at home without reading the paper. So, about 10 years ago, we proposed that this technique could be used to understand better how bacteria behave. And this is extremely important because, as you are probably aware, bacterial infections are becoming one of the major threats to human health. And so if we are able to understand better how bacteria behave, we should be in a better position to decide how to fight against these infections. And so in 2011, I obtained an ERC grant to study the communication between bacteria in a non-invasive manner by using surface-enhanced Raman scattering. So the idea is bacteria come close to each other. They interact with each other to really know how many individuals are forming a colony. And until they have enough individuals in the colony, they will not start a certain action, like forming a microcolony or a biofilm. And the biofilm is really determining also the start of infections. Now, this is based on a mechanism that is called quorum sensing. So the bacteria are releasing certain metabolites, which are selectively recognized by proteins that are located at the surface of other bacteria. And then, depending on the number of individuals, depending on the concentration of these metabolites, the bacteria will decide what they do next, whether they run away, or they form a biofilm, or they do something else. However, the detection of these molecules is not so easy to do without affecting the behavior of the bacteria. And so what we proposed was to design the most efficient Raman enhancing system to really place it underneath the bacteria colony and read what they are talking about. And so theoretical modeling told us that this type of assemblies were very effective because in every plane between two layers of our nanorods, there would be this very high array of hotspots where the signal would be largely amplified. And so, yes, we went on and designed this type of systems. This is, again, electron microscopy done in a slightly different way, where we can see the inside of these multi-step towers of nanorods that are really arranged exactly in the same, almost exactly in the same way as theory was telling us to do. And then with this, we could create arrays of these very well-ordered supercrystals made of nanoparticles. And then by illuminating with a laser beam in the presence of bacteria, we could actually start to see the signal of these metabolites, which indicate that the bacteria are talking to each other. And we can do this from very early stages of development and proliferation of the bacteria. So we would be in a good position to really understand this interaction and communication mechanism from the very beginning. Another advancement in this work was that we were able to implant this type of materials behind the skin of a mouse and still read with micromolar precision the presence of these molecules that would indicate, for example, the start of an infection after surgery. This is one of the potential applications of this technique. Now, after ending this project, although we are continuing our work in collaboration with our colleagues at the University of Vigo, we decided to go for a different idea and apply for a second ERC grant, which would focus on trying to help fight cancer. I don't need to explain why it is important to fight cancer. Our approach was actually to decide we are going to focus on breast cancer and melanoma because these are types of cancer which are closer to the skin. And so with our techniques and using infrared light, we would be able to penetrate the skin and study this type of diseases in vivo. But anyway, so what we proposed was, again, using surface-enhanced Raman scattering, but in this case, to build three-dimensional systems that reproduce the structure of a tumor, and then try to read the metabolism that is changing and this characteristic of cancer, while at the same time, we are encoding nanoparticles that will label cells so that at the same time, we can really see where every cell is located and how they are changing over time during the growth and expansion of the tumor. So this was very ambitious, and we have really progressed quite a bit. I'm going to show you very quickly. So this is one of the first experiments where we were designing a slightly different substrate with what I showed you earlier. Here we are using smaller arrays of nanoparticles, which again become very efficient because of the precise ordering in a two-dimensional manner. And here, by irradiating with a laser beam, we are going to study the metabolism of cancer cells that overexpress an enzyme that is called IDO. And so in this particular enzymatic process, the metabolite tryptophan is converted into kynurenine. So the first thing we do is to study the Raman spectrum of the two metabolites and identify the peaks that would be characteristic for each of them. And after doing that, we can culture the cells, extract the supernatant, and then start measuring over time. And we can really monitor the decrease of this peak and the increase of this other peak, which is reflecting the metabolism of this specific cancer type. Now, this is a very simple way of doing it, because actually in this type of spectra, you have much more information than what you see in one particular peak. And this is why more recently we started to do more complex data analysis, starting from standard principal component analysis, a very well-known statistical method, but also by using more modern machine learning techniques combined with a microfluidic system where we can obtain thousands of spectra from our systems that we know, in this case applied to cell death mechanisms, also very important for fighting cancer. And so with these thousands of spectra, we train our theoretical model, and then after that, we can really analyze the data of new systems and identify the death mechanism. In this case, for example, we are distinguishing between necrosis and apoptosis, which are different mechanisms which depend on how cells are dying after the effect of external stimuli. Now, this is very important. Now, the next thing I mentioned, we can label cells. Of course, if you are close to the biology field, you will tell me that you can already do that by using fluorescently labeled antibodies, for example. And this is true, but with fluorescence, you have some limitations, for example, related to the breadth of the emission peaks, so you cannot really use so many labels at the same time. And also, different labels will require illumination with different lasers. In surface-enhanced Raman, you can have as many of these barcodes as you like by simply changing the molecular structure of this Raman reporter that we link to our nanoparticles. And then you will have a very intense signal, which is characteristic of each of the codes. And again, you can obtain all of these signals by illuminating with the same laser beam. So you can analyze all of the tags exactly at the same time. Additionally, these signals do not bleach, they do not change with time, they are not affected by the illumination. So you can also study these processes in longer time scales. Let me just show you very quickly an example. This is a 2D multiculture of 5 different cell types, each of them coated or labeled with one of these tags, and we can see how they are evolving and moving over time. We can identify at each time point exactly the location of each cell. And nowadays we can do this also in 3D, so we can study multi-layers of cells labeled with different tags and identify how they are also evolving over time. Now, we wanted to go into something a bit closer to what happens in a human body, because the interactions between cells in a 2D culture and inside the body are quite different. First of all, because in the body we do not have a substrate on which the cells are attaching. Second, the cells need to really talk to each other and need to be also accessible to all the nutrients and the components of the extracellular environment that we have in vivo. And so there are typically these 3 different levels of approaching what happens in a human, from the 2D cell culture to the animal model. We wanted to stay in between and apply so-called 3D bioprinting to create scaffolds that can be used to culture the cells in 3 dimensions and somehow simulate what happens in a human. And indeed, we were able to do that. We purchased a commercial 3D bioprinter that is working mainly by extrusion in different modalities. So we have to design an ink that will be compatible with our nanoparticles and with the cells, mainly using polymeric components that will form a hydrogel, where the nanoparticles can also be embedded. And then by using the right combination of these materials, we obtain a fluid that can flow through this system and then will become rigid when going out of it, so that we obtain these three-dimensional structures containing in each of these lines the nanorods well dispersed. And then we can also culture the cells that were also labeled with our nanoparticles, and we can obtain these three-dimensional images where within the scaffold we can do the detection of the metabolites, while at the same time we're identifying the position of the cells, which are here shown with the red label. So we are already in a good position to study what happens in three-dimensional models combined with the identification of the cells. Very quickly, an example of how this can be used for drug evaluation. We print one of these scaffolds in a microfluidic chip. We fill it with happy cells, which are labeled here in green. This shows us that they are alive within a hydrogel that is filling the holes of the scaffold. And then we use a drug molecule that can act only upon illumination. And then we can monitor at the same time the diffusion of the drug through this system by simply measuring the Raman signal of the drug in the scaffold, and at the same time doing the cell viability test by looking at alive or dead cells. So we can correlate the red dots here that will indicate the death of cells with the diffusion of the drug. So by using different drugs, we will be in a position to decide which one is acting faster or more effectively. Still, these systems do not really resemble the full structure of the tumor. So let me show you very quickly. In the real tumors, we have something like this. We have typically a core with a very dense array of cancer cells, which is surrounded by so-called stroma, which contains different components like collagen and a few other things, and also different cell types, like fibroblasts or endothelial cells. And all of this comprises the environment where the tumor will want to expand. And this is what we really need to understand. And so recently, we were really working into obtaining the most similar system to what we are seeing here. And so in order to obtain something that really resembles what happens in an animal, my students were working very hard taking tissue from dead porks, from the mammary glands, to extract the pork-related cells and DNA components, so that they obtain a hydrogel that can be formulated as an ink and then be used to grow the human-derived cells in a way that they are behaving as in the human. And so actually you can see here how these cells are spreading very well, which is not so common by using the commercially available hydrogels that we typically have access to. So after this hard work of developing these inks, we were in a position to try to simulate this morphology by using, again, the 3D bioprinter with two different formulations, one for making the cancer core, one for making the external layer. And so this is one of the examples of the systems that we were able to fabricate, where we are labeling different cell types, again, in different colors, and which reproduce really something very close to what happens in a human tumor. And with this type of systems, we were also able to evaluate different drugs which are known to be effective as anti-cancer drugs, including tamoxifen, which is known to be effective for certain breast cancer types and not for others. For example, here we evaluated the efficiency for two different cancer cell types, NCF7, which is a more standard cancer breast cancer type, and tamoxifen is effective, and you see that by increasing the concentration of the drug, you increasing the level of cell death, while for these other cell types, the so-called triple-negative breast cancer, which is more aggressive but also more selective to certain population, this drug is really not so effective. So again, now we are much closer to deciding what will be the appropriate treatment for a certain group. So our ambition is really by using the knowledge that we had from the basic science of making, characterizing, and understanding nanomaterials to be able to contribute to the transition from the current medicine where one solution treats everything to a personalized medicine where we are diagnosing different population groups and then applying the appropriate treatment. Nowadays, this is done already by using genomic sequencing or proteomics. We are dreaming with a future where the SERS test will be part of the evaluation of the patients in order to decide this. Before I finish, I would like to spend 2 more minutes to acknowledge the participation of so many people who have worked in my labs over the years. You will see first PhD students and then postdocs, and we will quickly fill the screen with people who have contributed to provide ideas, to carry out the experimental work, to the discussion, to writing articles and patents, and also to collaborate with other groups everywhere in the world. And so, yes, thank you to all of these people. I would also like to acknowledge the collaboration with so many other people also at EMAT and other departments of the University of Antwerp. Again, this will not be so crowded, but still, over the years, we have collaborated also with a big number of people in this university, and we are so proud and grateful that I also wanted to acknowledge this at the end of this lecture. So with this, I finish my masterclass. I'll be happy to answer questions. Thank you very much.
Okay, may I invite you to join me around the table? So, Professor Liz-Marzán, thank you so much for this very interesting masterclass and for sharing these impressive results with us. So it's really nice to see that blue sky research can lead to such beautiful applications. So please, you can join me here. And then I think it's time to move on with the questions. So we have an online audience, so I hope to receive questions here at the iPad. And of course, we also very much welcome questions from here. So you can simply raise your hand. I think Wouter is there to give you the microphone so we can all hear you loud and clear. See already congratulations coming up here on the iPad. I see there is one question here coming, so I will start with this question, and then later I hope to see hands being raised. Can self-assembled nanoparticles also be used for meta-lenses?
The answer is yes, but this is not really my field of research. But yes, metamaterials are one of the most popular applications of metal nanoparticles. So the combination of plasmonics in specific arrangements, and of course, self-assembly is one of the methods of fabrication that could be used with our types of materials. So the combination in specific arrangements will also interact with light in very different ways to what I showed you here. And one of the popular applications of this would be the so-called invisibility cloak. So indeed, by the arrangement of these materials in these very well-designed patterns, might really promote the deviation of light in a way that will be still transmitted, but hiding part of the image that you have in front. And this is the concept behind metal lenses that is described in the question.
Okay, thank you. Questions from the audience here. So please raise your hand. I see a question there.
Thank you, Louis. Wonderful talk, as always, and congratulations with the honorary degree. One very general question. To apply SIRS in a diagnostic setting, it largely depends on the uniformity of the SIRS enhancement. Can you elaborate on how far you have pushed that with these assemblies, how uniform the SIRS enhancement field, if you want to call it like that, is really established in the concepts that you've shown here?
Yes, this is of course a very good question, and thank you, Johan. Right, I think that there are multiple answers for this question, because it all depends on how accurate you want to be and what sensitivity you need. And so in the 3D scaffolds that I showed, we are not detecting very low concentrations. We are typically around the micromolar regime. And therefore, we aim at having a uniform distribution of the nanoparticles within the hydrogel in the scaffold, so that we can obtain a signal everywhere, even if it is not the highest that we could achieve. And so that will tell us already whether there is a certain metabolic process going on that we can identify through this data analysis that I described. And the signal from the labeled particles in the cells will also be strong enough, and just by having the signature, we should be able to tell where the different cells are. Now, if you want to go to very high sensitivity, then it's not only the uniformity of the substrate that you have to worry about. This is important, of course, to obtain reproducible signals from different spots. But also, you need to make sure that what you want to analyze will be approaching and staying long enough on the substrate so that you can obtain a reliable signal. And this is not always possible. For example, in the field of environmental monitoring, this is a real problem. Not only because there may be many other substances that can be around that can contaminate the signal, but also many of the substances that we are really interested to detect are not very prone to absorbing onto the metal substrate. And therefore, you have to design also ways to capture those molecules and then do the identification. So indeed, for every application, there would be a sort of tailoring of the technique and the material so that you can apply it reproducibly.
Thank you.
It's a little bit in the same line. You want to use it later as a diagnostic tool, but then SIRS is a very special technique. You need specialized people. The setup is not so easy to handle. So how do you see that in future?
Yes, I would disagree slightly with that, because again, if you really want to do SIRS research or go to very high sensitivities, you do need a specialized equipment and very detailed data analysis. If the application allows the use of portable Raman instruments, then everything becomes not only much cheaper, but also much easier to use. And the data analysis can be automatized. So I think that indeed one of the advantages of the surface enhanced Raman, again, for the type of analytes that will like to bind to the substrate, is that it requires almost no sample preparation. And so you could simply use your sample, dip it on the substrate, do the measurement, and get the data out. This is, you could have said the same thing about light scattering 30 years ago, that you would really need very specialized people and equipment to do the analysis of particle size. Nowadays, we have closed boxes. Very often, I'm even afraid that people are obtaining results that do not correspond to what they see. But in most cases, this is reliable and can be used safely. So I think that in the near future, this can be exactly the same for SIRS.
Thank you. Are there more questions in the audience? Please, Professor De Veter.
Thank you. I had a question concerning the surfactants that you use. So you use them to control the growth of the nanoparticles, but for the applications, at what point do you need to get rid of them, and does that affect the stability and the functionality? Can you comment on that, please?
Yeah, of course, a very, very common problem that we have to face, and that we indeed learned how to solve over the years. And there are, again, different answers depending on what you want to do. So very often you would like to get rid of the surfactants. If you are using a high amount of the surfactants, they can also be strongly cytotoxic, so that would affect your application. So what we do is, first of all, to do as much cleaning as we can so that we actually at least get rid of the surfactant in solution while preserving the colloidal stability of the material. And then, for example, in the fabrication of these nanoparticle, including hydrogels, you can simply adapt the concentration of the surfactant to the compatibility with the hydrogel formulation. And then within the hydrogel, the surfactant will not be toxic anymore. And because we made sure to have the minimum possible, then the molecules that we want to detect will still be able to reach the surface of the particle and the signal be amplified. So again, for every application, you would have to design a technique or a path to solve the problem of the surfactants if there is a problem. For some applications, you don't even need to worry about that. But it is very relevant. Thank you.
Thank you. Here on the chat, I see there are many congratulations, especially also from the group in Utrecht, so well deserved. Another question is how to translate a hotspot functionality to applications deeper in the body. Are there other wavelengths that could provide this?
Yeah, if you want to use this type of sensing or treatment deep in the body, I'm afraid that the only way would be to use optical fibers and be a little bit invasive. So you will have to be invasive already to introduce the nanoparticles, and then you will have to be invasive to introduce the optical fiber. And this is why we are mainly proposing applications closer to the skin. But I'm aware that there are already in the US clinical trials to use this type of materials for treating prostate cancer, which is already not so close to the skin, and of course they are using optical fibers to induce that. So I think it all compares to what you are going to replace. So if you're going to replace surgery with introducing an optical fiber, I think there will be a good reason to do that. Again, it all takes different solutions for different problems, but there is a solution for it.
Okay, thank you. Question in the back.
Hello. Hello. So thank you for the clarity, first of all. This is quite rare nowadays, and your lecture was extremely clear and interesting. So about the surfactants and the removal of surfactants again, would the decoration of the nanoparticles with thiol, gold-bound, organic monolayer introduce practical problems, or would that also be a possibility for the removal of the surfactants?
Yeah, this is one of the major methods to actually get rid of the surfactants. So again, depending on what you want to do with the particles, you will choose a different thiolected molecule, but this is really done every day. So indeed, because thiols are very strongly binding gold surfaces, the use of molecules containing thiol groups is one of the popular methods to modify the surface chemistry of these materials. Thank you.
That's a question in front here.
Your work is a beautiful explanation of the importance of multidisciplinary research.
Yes,
but in order to do that, you need to communicate between scientists from different levels. And as I see it, the financial systems, through departments, to faculty, to individual professors, is more in towards a vertical education than horizontal. I see it in all cases I've seen. What is your meaning on that?
Well, Of course, in terms of education, there is always the compromise of going deep enough that you really understand one subject and being broad enough that you are prepared to tackle different problems in this modern world of multidisciplinarity, which we may also say gets back to the origin of the philosophers who were also doctors, chemists, physicists, and so on. Exactly. So Faraday actually did not study, so he learned on his own, but anyway. In my particular case, I had the privilege to be able to continue learning during my whole career, and also to obtain enough funding that I could recruit people from different education systems and different expertises. So we can really combine in the lab people who have studied chemistry, physics, pharmacy, biochemistry, medicine, bioengineering. And I think it is only the combination of this team that allows us to present the work that I showed you today. I would not have been able to help in the lab to build these complex cellular systems unless I could recruit very skillful postdocs who are really driving this type of work. So I completely agree that we need multidisciplinarity, but I also think that unless we understand well what each person is doing, it will be difficult to do it properly. And this is also something that Faraday was saying. Before you publish, you have to finalize. And you can only finalize if you understand the reasons for what is happening.
And there is another question here in front. I see 2 questions.
After working on artificial tumors, are you going to move to artificial organs?
I think that organoids require many more components. So I don't want to say no. I already said many years ago that I would never do bio, and look at this. I don't want to say no. In fact, close to my lab, so people who were working with us and who are now starting a new lab, they are actually going in that direction. They are formulating artificial arteries to be incorporated into organoids. And so we may be collaborating with them or not, but it is a direction that I find very attractive, but I don't want to predict whether my future will be there or not. But I find it really fascinating. Thank you.
As a biologist, you know, there's obviously a lot of very interesting things that you could label and follow in real tissues rather than artificial tissues. And the main thing that immediately springs to mind when I hear your expose of you know, dipping samples and labeling them is to what extent, you know, do these microparticles you know, go right through the cells, or are you just looking at outside labels?
Right. So when we are labeling cells, they typically end by endocytosis. So they are inside vesicles inside of the cells. And this is an advantage for us because that will not really affect very much the biology of the cell. It's a problem if we wanted to use them differently, for example, to do intracellular detection. If we wanted to do intracellular detection, we would like the particles to get out of the endosomes and be accessible to what we want to detect. And so there are also methods to do that, which we are not yet exploring, but that really might be in the planning.
So that then refers mainly to, I suppose, animal or bacterial cells. I'm a plant biologist, and they don't do much endocytosis. Am I really disadvantaged there?
Well, I don't have any experience, but there is quite a lot of activity on the interaction of nanoparticles with plants. We recently published an editorial in ACS Nano related to nano and plants, where maybe you can find solutions or ideas to go in the direction that you're interested. And so I cannot really give you a detailed answer how the nanoparticles go through the plant cells, but there is diffusion, at least through the capillaries in plants. And I think, yeah, there would be definitely plenty of room to study also interactions and ways of affecting the biology. Yep, thank you.
Okay, I will take one last question from the online audience, from Ghent. Thank you. My question is related to the nanoparticle synthesis steps. Could you please elaborate more concerning the break symmetry step in the synthesis? So what do you mean by breaking the symmetry?
Right, so breaking the symmetry simply means that we go from spherical to anisotropic. And this, if you think about it, is difficult to conceive in something like gold, where the crystalline structure is almost isotropic, and the energies between the standard facets are very similar to each other. And so breaking symmetry means that you are somehow affecting the energy of one particular facet so that it will develop, and then you will end up with an anisotropic morphology. And this is something that we really learn to control again through the work of many students and postdocs who are providing all of the work and many of the ideas.
Okay, thanks a lot. Thank you. Thanks a lot for all these interesting questions and answers. I'm sure that we could go on for a while, but since there are other important things to do now, I suggest that we take a seat.
Okay, thank you.
Thank you so much, Professor Liz-Marzán, for your inspiring masterclass. And of course, there are other very important things to do right now, because it's time to confer the honorary degree on Professor Louis Liz-Marzán. Nominator Professor Sarah Bals will deliver the laudation before the official conferral, and afterwards the honorary doctor can give his own word of thanks, of course.
The first time we met Louis was in February 2011. At that time, Staffan Tendelo was involved in a new European project called European Soft Matter Imaging, ESMI. It was a topic that was far outside of our comfort zone. And Staff and I, we went to the kickoff meeting in Düsseldorf, not really knowing what to expect. As the day of the meeting continued, I was feeling more and more concerned because I had no idea how we could be part of this community using transmission electron microscopy. That was until Louis stood up and gave his presentation in which he discussed plasmonic metal nanoparticles. It felt as somebody had literally switched on the light. Staff poked me and whispered to me, we should go and talk to him. And that was the start of a very close and successful collaboration. Louis, I have deep respect for all of your scientific achievements. And it is impossible for me to understand how you are able to continuously challenge all of your collaborators to go beyond the state of the art. You have raised the bar and you have pushed the community because of your unique vision on science, but also because of your personality. You have such an amicable way of doing science while at the same time being one of the most critical scientists I know. Collaborating with you has opened doors for a large number of researchers at our entire laboratory. You have inspired them to develop novel characterization techniques, which have enabled many more high-impactful publications. In addition, I cannot count the number of times people have approached me at conferences saying, I'm a friend of Louis. And it is striking that all of them, and many of them are here from all over Europe, said, I am a friend of Louis, instead of I'm a collaborator of Louis. One of the things that is very special about you is how you have remained so approachable. Despite all of your activities and responsibilities, you are always willing to collaborate, always willing to help, whether that is by preparing the most complex particles, by helping to understand a difficult growth mechanism, or simply by providing some good advice. Because of this, you have impacted the careers of more researchers that you can probably imagine. And in order to convince you that you are gold to us, we have prepared the following message.
From nanorod to nanotriangle, you always challenge us with the most interesting particles by far. Today we want to say congratulations, because today you are the star.
Gold, always believe in your soul. You've got the power to know you're in the...
Congratulations, Louis. Well deserved.
Your work inspires many of us. Congratulations on your well-deserved honorary doctorate. Be proud of it, because you've earned it.
Hi Louis, congratulations on receiving the honorary doctorate. It has always been an absolute pleasure investigating your nanoparticles. So well deserved, big congrats.
Hi Louis, congratulations for your award. I hope we'll be able to continue collaborating together in the future.
Dear Louis, congratulations on receiving an honorary doctorate.
Professor, congratulations on your great achievement.
Hi Louis, many congratulations for the honorary doctorate, and many thanks too for all the nice collaborations.
Congratulations.
Felicidades, Luis.
Always believe in your soul. You've got the power to know you're indestructible.
Dear Louis, congratulations on your honorary degree from the University of Antwerp. I wish you more fruitful collaborations and impressive results.
Hi, Professor Liz-Marzán. Congratulations on receiving an honorary doctorate from the University of Antwerp.
Congratulations, and let's hope for a bright future with many more nanostars.
Congratulations, Louis.
Dear Louis, when working on the samples from your group, you're always sure that it's gonna end in a nice and fruitful publication. Therefore, I would like to congratulate you on your honorary degree and wish you even more nice and impactful publications and collaborations.
Hi Louis, congratulations with your honorary doctorate. See you soon, bye.
Many congratulations, Louis.
Congratulations.
Congratulations, Louis.
Well deserved. Greetings from Utrecht.
Always believe in you are gold. I'm glad that you're bound to return. Something I could have learned. You're indestructible.
Hola Luis, felicitaciones por el doctorado honorífico. Un besote de París.
Hey guys, have you guys all heard? There's a new piece of news out. Luis has been given another premiere.
Ah, the one from the National Academy of Engineering?
No, not that one, Gail. A different one.
¿El premio Luz Español?
Neither. Well, no, he also has that one, but it's not that one.
It was more for the member of the Royal Academy of the Sciences of Spain.
He also has that one, but it's not that one. I bet Vigo, do you guys know which one it is? So Vigo, you guys don't know either. Well, I'm going to have to tell you all then. So he's now got an honorary doctorate from the University of Amberes.
Wow!
Sorionak, Luis, musu pilo bat.
Hola, patrón de embarcación de recreo, ¿cómo estás? Enhorabuena por ese súper merecido honoris causa. Estoy deseando salir aquí al mar contigo de nuevo, a ver cuándo podemos hacer. Te queremos mucho, enhorabuena, felicidades.
Hola Luis, enhorabuena por tu distinción como doctorado honoris causa por la Universidad de Amberes. Espero que lo estés disfrutando mucho, te lo tienes merecido. Un abrazo.
Sorionak, Luis, que por fin eres doctor, ¿eh?
Hola Luis. Bueno, que ya me han dicho que te han dado otro premio, así que nada, uno más para la colección. Enhorabuena y a recibir muchos más. Un besito desde Dubái. ¡Mua!
For all these reasons, I request that the Rector honor Professor Dr. Luis Manuel Liz-Marzán with an honorary degree from the University of Antwerp.
Thank you. A special thanks to the Faculty of Science for this fantastic presentation at the end. Dear attendees, on the recommendation of the Faculty of Science and by decision of the Executive Board, the University of Antwerp confers upon Professor Liz-Marzán, Center for Cooperative Research in Biomaterials, CIC Biomaster and University of Vigo, the degree of Doctor Honoris Causa in Sciences, in recognition of his expertise in the synthesis, characterization and application of plasmonic metal nanoparticles. Congratulations, and hereby I confer you the medal of the University of Antwerp, which is bright on your fantastic gown, and also one more diploma in your collection. Congratulations. Thank you very much. Okay, and hereby I invite you, please.
Well, I usually get emotional during my speeches. Today, they managed to make me emotional before starting the speech. By the way, I'm gonna give it a try. Don't worry if I stop in the middle. Eigenlijk, ik wilde enkele woorden in het Nederlands te geven. Ik ben helemaal trots om deze grote eer te krijgen van de Universiteit Antwerpen. Ik moet de rector en de hele universiteit en de faculteit wetenschappen te danken om dit grote eer te gunnen. En natuurlijk professor Sarah Wals, om al die dingen dat zij hebben al gezegd. Maar ook voor een geweldige vriendin te zijn. En ik hoop dat dit eindigt niet hier. En wij voor vele jaren nog meer discoveries kunnen maken. Anyway, I already mentioned the students, postdocs, technicians, collaborators. It's really they who actually make the materials that are studied here. And I should say, for me, it's very easy to say, yes, we are going to do what you are asking. But then it is them who have to do it. So it's really, I'm really privileged to count with so many good friends and collaborators at all levels. Of course, I'm really grateful to those colleagues and friends who made the effort to come here today, and also to all who are watching online, and including, of course, family. And I have here my closest family, and they always make the effort to come for every of these events, and this was a very special one, so they are coming from everywhere in Europe. But I know that there are other family members also watching online, and I want to send a special hug to my brother Eduardo, and he knows why. Dank u wel, iedereen, and thank you, and I'm really happy for this recognition.
I'm supposed to just guide you from the stage. That's it. No, I don't think so.
Nee. Er toch nog foto's? Ja.
Maybe,
you can take your seats. I think the pics are okay, I guess. I would like to finish this morning by saying a few words of thanks myself. And of course, I'm gonna do it in Dutch because, yeah, you have a great accent. Dank je wel, professor Liz-Marzán, voor het ongelofelijke werk dat je doet, voor de heel interessante masterclass. En natuurlijk om deze graad, dit eredoctoraat, te accepteren. And next to every successful man is a strong woman, or another man, but in this case, it's a woman. So many thanks to you, Charo Pastero Gomez, also for joining our ceremony this morning. And we have a small but delicious and typical Belgian present for you. Thank you to Rector Herman van Goetem, to Dean Gerrit Beemster, to nominators Sarah Bals, and of course also thank you to the Faculty of Science and to the University of Antwerp. And of course I also like to thank you for all joining us this morning here in this room and also online. And have a nice day, and I hope to see you again in 2024.